Well, welcome back. This is part 2. This is where we start to get a bit more dynamic. But first we need to talk about a bit more detail about the structure of skeletal myocytes in terms of working down from the big muscle down right the way down to the sarcomeres and the myofilaments. Then we'll start to put it together and we'll talk about excitation, contraction coupling. First of all, we'll talk about excitation. Excitation involves action potentials in both the neurons serving the myocyte, which are then continued in the myocyte. So we're going to need to learn about action potentials. We're going to need to learn about synapses and then how that causes contraction. So we'll be looking at how excitation causes the release of calcium within the skeletal myocyte. And then we'll talk about contraction, which is a really amazing process involving the binding of calcium to one myofilament, the interaction of another myofilament with another myofilament, a bit of ATP thrown in, and we have this whole process that contracts the myocyte and consequently, if you contract enough myocytes, the whole muscle. So in various diagrams, you will see skeletal myocytes called muscle fibers, and that's exactly what they mean. But correctly, they are skeletal myocytes. Each of those myocytes, as we said before, extends the full length of the muscle. Because it's so long, and because it needs so much ATP, so much remodeling, it has lots of nuclei. So skeletal myocytes are multinucleate. They're the only normally occurring cell in the body that is multinucleate. However, in terms of a muscle, not all of the skeletal myocytes will be excited and will contract at the same time. If you think about it, if they did, we have no regulation, no graded responses. For example, if we wanted to pick up a pen or something, a piece of paper, something very light, we don't need to contract the whole muscle. But if you're going to pick up a heavy box, you might need to contract the whole muscle. So this stimulation, this excitement and contraction within specific myocytes within the muscle gives us these graded responses. The other thing it does is prevents muscle fatigue. What happens is a few will contract for the initial effort. If that effort is sustained, those myocytes then have a bit of a rest and other myocytes come into play. It's a bit like if you're into farming, fallow fields. You rest a field for a year so the nutrients can build up again before you plant in it. So this prevents muscle fatigue. And if you think about it, if you are lifting that heavy box, you need to contract the whole muscle, all of those myocytes within that muscle. You can't sustain it because you don't have any resting in reserve. The contractile myofilaments that we talked about, remember actin and myosin, within the myocyte are arranged in these sarcomeres that we talked about before. It's the sarcomeres, and you can see there in that diagram, that give the striations. Because they're all lined up, and all the myocytes are lined up, we get the striated tissue. Sarcomeres are short sections, end to end, full of the contractile myofilaments. The sarcomeres are repeated along the myocyte within specialist organelles that these types of myocytes have, called myofibrils. So within a myocyte, you have these very, very long, going the full length, organelles called myofibrils. Those myofibrils are then divided up into sarcomeres. And within the myofibrils, divided into sarcomeres, are the myofilaments. Each sarcomere will contract along the whole length in every myofibril and in every myofibril within that myocyte. So although not all myocytes will be contracting, within a single myocyte, contraction will occur in every sarcomere in every myofibril at the same time, simultaneously. And we talked about that before in terms of other types of muscle. In this case, it's happening within a single cell, a single myocyte. So it contracts, each myocyte contracts syncytially. This is a true syncytium because it's within one cell. In terms of when we talked about smooth muscle and cardiac muscle, contracting as a tissue simultaneously, syncytially, that was a functional syncytium. So let's work it down from our muscle. So there you can see a bone. So yes, a bone is what type of tissue? Yeah, it's connective tissue, isn't it? Specialist connective tissue. Then we have a tendon, also connective tissue, but in this case, proper connective tissue. And then from that, of course, we have the muscle. The muscle is full of myocytes. And around those myocytes, you can see that they're organized into bundles. And these bundles are wrapped in more connective tissue to form fascicles. Fascicle is just a bundle. We'll come across the term again in a different tissue. Within those fascicles, we have the single myocytes all bundled together. Around the myocytes are capillaries. We need a good supply of blood to myocytes. Within each myocyte, as we said before, we have these specialist organelles all packed in. And because they're packed in, they push all those nuclei out to the edge. So most of the inside of the myocyte is full of these myofibrils. So within myocytes, we have the myofibrils, and these are the specialist organelles. Those are then divided up into sarcomeres, into sections called sarcomeres. We'll look at sarcomeres next. Within those sarcomeres, we have our myofilaments. And there you can see right at the end sticking out an actin myofilament and a myosin myofilament. And it's the interaction of those myofilaments that gives us contraction. As we said, so each myofibril is divided into sarcomeres. In the middle of the sarcomeres, we have what is known as an M line, M for middle. If we look in detail at a sarcomere, you can see the M line in the middle. The blue lines in this diagram are actin. Actin is called a thin filament. And notice that actin doesn't reach across the M line. There is a gap in the middle. This is known as the H zone. But The big pink spiky fibres there are myosin. We'll find out in a minute why they're spiky. They are known as the thick filament. And you can see that they sit amongst the actin. Either end of the sarcomere, we have something called a Z line. We're not going to worry too much about that, but basically you have these elastic fibres there that they permit contraction and relaxation once there's no more excitation going on. So the thin blue myofilaments there are actin. And when relaxed, actin doesn't reach the M line. We have this gap in the middle. The thick pink myofilaments are myosin. Myosin, the little spikes that you see sticking out there, we'll look at those in a bit more detail. The little spikes are heads sticking out in all directions from the myosin filament. If we look at it in cross-section, and we rarely do because if you're trying to explain how this interaction happens, you need to do it in 2D, otherwise it would get very confusing. But remember that if we look at it in cross-section, what you can see is that each myosin filament within the sarcomeres is surrounded by 6 actin filaments, because these myofibrils, remember, are round. So in 3D, if we take a cross-section, we can see that they actually are bundles of myofilaments. So where we have our 2D diagram there, we would have another actin filament, for example, in front of that myosin and behind it, interacting all the way around the myosin. So, okay, let's talk a bit more about actin, and we'll introduce another myofilament. The thin actin myofilament is made up of actin molecules. These molecules are arranged in a double helix. It's a bit like taking 2 strings of beads and twisting them. So we have this double helix of actin molecules. But then twisted around that, we have a very fine myofilament called tropomyosin. Not to be confused, of course, with myosin. This is tropomyosin. Tropomyosin wraps around the actin filament. Every 7th actin molecule, it's secured by something I'm sure you've all heard of, troponin. So we have this troponin complex of 3 subunits that secures the tropomyosin to the actin. If you need to try and remember troponin and tropomyosin and which one is which, I always say to students, troponin is a short word. So therefore it sums up this little small complex of 3 subunits. Tropomyosin is a long word. So that's the long filament that wraps around, secured by troponin. As I said, the troponin complexes have 3 subunits. One of these subunits has 2 calcium binding sites, Ca++. Calcium is a double cation. And remember, when we're writing this, we always use the symbol Ca++. Otherwise, you would have to write calcium double cation. And no one wants to have to do that. Ca++. You may see it written as Ca2+, and that's perfectly correct too. So when calcium, Ca++, binds to the troponin, remember 2 per troponin, the troponin then moves tropomyosin into the grooves on this helix of actin. it undergoes a conformational change whereby it falls into the grooves on actin. What this does, it exposes myosin head binding sites on the actin molecules. Each of these actin molecules has these myosin head binding sites. Normally, in relaxed muscle, tropomyosin sits on the top of the beads, if you like, the apex of this helix, covering these binding sites. When calcium binds to troponin, it moves tropomyosin into the grooves of the helix and away from those myosin head binding sites. Those myosin head binding sites, when calcium is bound, are exposed. Once they're exposed, contraction can happen. So therefore, the release of Ca++ calcium to the myofilaments is the stimulus for contraction, even though the calcium only prepares actin really for myosin to bind to it. It's the start of contraction, the initiation, if you like. So myosin then. Myosin is that thick filament that does cross the M line. We said it has these myosin heads. Those heads are stuck up on little twisted tails. Normally, in a relaxed state, they're floppy. They're in a low energy state and they're floppy. These myosin heads also carry ATPase. So what does that tell us? Yeah, it tells us we are going to need to hydrolyze ATP for contraction to happen. So to bind to actin, they need, first of all, the myosin head binding sites on actin to be exposed by the binding of calcium to troponin, which moves tropomyosin away, and they need a supply of ATP. And it's this interaction of the myosin heads and actin that we're going to talk about later that causes the contraction in each sarcomere. So, before we talk about contraction, let's get excited. This has to come first. It comes first because this whole process is known as excitation, contraction, coupling. It's a combination of events that ultimately will lead to that myocyte contracting. So contraction requires excitation. Skeletal muscle excitation starts up in the primary motor cortex in the brain, that slice alongside the sensory cortex that is a motor cortex. Obviously, this is an efferent pathway because it's coming out. Remember, we said all motor neurons are efferent. But initially, we have to generate the action potential, and that happens within the primary motor cortex. The action potential then travels all the way down the spine via an upper motor neuron and then travels out from the ventral horn of the spinal cord to a neuromuscular junction via the lower motor neuron. This is where the term upper and lower motor neuron comes from. Upper motor neurons are within the central nervous system, run down to the correct spinal level, and then come out via a spinal nerve as a lower motor neuron. all the way down to the neuromuscular junction. The depolarization of that action potential is then passed over to the myocyte via the neuromuscular junction. The depolarization carries on along the membrane, the sarcolemma of the myocyte, and it stimulates the release of calcium. And as we found out before, calcium then binds to troponin, moves tropomyosin out the way, and off we go with contraction. So excitation really starts in the motor center in the brain, crosses over the neuromuscular junction, and results in the release of calcium. That's excitation. So it involves action potentials, which means we're going to have to talk about action potentials. We'll come back to it in neurophysiology, but hopefully this will be fairly straightforward at this point. Action potentials are a wave of depolarization that moves along a plasma membrane in any excitable cell. So those excitable cells, generally we're talking about neurons, or we're talking about myocytes. They are also excitable. So let's talk about action potentials. Action potentials, if you remember when we talked about resting membrane potential, we talked about the membrane being more positive on the outside compared to the inside. We had lots of sodium Na+ clustered on the outside of the plasma membrane, making that quite positive compared to the inside. In an action potential, we get the reversal of that, so we become more positive inside, which is then later restored following this wave of depolarization. So the polarized membrane, remember, resting membrane potential is when the membrane is polarized. So our polarized membrane at RMP is first polarized, and then repolarized. There is another bit in between, but we'll talk about that in a minute. When depolarized, it is more positive inside the membrane, which of course is the opposite of being polarized at RMP. This wave of depolarization moves in one direction. There are various mechanisms to ensure this that, again, we'll talk about later. The wave of depolarization moves in one direction along the plasma membrane in irritable cells, i.e. neurons or myocytes. Of course, if we're going to change, we're going to cause depolarization, we're going to change the potential, completely switch it round, reverse it across the membrane, we're going to need to move ions. So an action potential involves the opening and closing of voltage-gated sodium and potassium channels. And it is important to keep clear that these are voltage-gated. They have voltage sensors. And it's voltage, or the electrical charge around them, that causes them to open. If you remember, our other type of channels are ligand-gated or mechanically gated. We will come across some ligand-gated channels when we get to the neuromuscular junction. Otherwise, they are all voltage-gated. Following this, the sodium-potassium ATPase pump, remember that one, restores resting membrane potential by pumping out 3 sodium and pumping in 2 potassium using ATP. So let's look at that in a little bit more detail. As we said, these are all voltage-gated ion channels, and they all have voltage sensors. The thing about voltage sensors is that they have a point at which they will open the channel, and that point is called threshold. So when we're talking about threshold, we're talking really about voltage sensors, and the threshold can be varied a little bit, but generally in terms of an action potential in a neuron, for example, or a skeletal myocyte, we tend to talk of the same figure, an average figure. So the voltage sensors cause the channel to open when they reach threshold. They reach threshold if there's an adjacent rise in voltage for any reason. Now, at this stage, don't worry about what that reason is. We'll come across lots of occurrences when that happens, and then we're off. Just for the moment, accept there can be an adjacent rise in voltage. Most neuron voltage sensors reach threshold at around minus 50 millivolts. And if you remember, say for example, neurons, resting membrane potential is minus 70. So this is getting, bearing in mind we're talking minuses, closer to 0. So it's increasing, it's going up on a graph if you like. So from minus 70, when those channels experience an environment around them that has a positive charge, they reach threshold when that charge gets to about minus 50 millivolts. When the channels open, ions, this is passive transport, so ions follow their electrical and or chemical gradients, and either, depending on those gradients, influx, move into the neuron, or efflux, move out of the neuron. So this is the point that we are going to depolarize and repolarize, depending which channel we've got open and what ion is moving which way. So, as we said, resting membrane potential, we have that accumulation of sodium outside the membrane. That sodium outside the membrane, remember, has a strong electrochemical gradient, and it wants to influx. But generally, at resting membrane potential, all the sodium channels are closed, so it can't. In terms of concentration, we also have more potassium inside the membrane than outside the membrane. This potassium can efflux and influx through the potassium channels. These are different channels to the ones we're seeing here. These are not voltage gated, they're just potassium channels. But that intracellular concentration stays constant through this equal 1 in, 1 out. Sodium, as we said, Na+, wants to influx following its electrochemical gradient, but generally, while the channels are closed, it can't. So, for an action potential to happen, there is an adjacent rise in the potential. So there is adjacent rise usually in intracellular sodium. So sodium moves in, it's gathering, diffusing through the cytosol, it's gathering around that voltage sensor on the channels. Once there's enough gathered around the voltage gated sodium channels, around the voltage sensor specifically, The voltage sensors, if they reach their threshold at minus 50 millivolts, they will open the channel, and our channel opens. When the channel opens, sodium, of course, now has an open channel, has a strong electrochemical gradient, so it influxes in a big flood. This, of course, has now reversed resting membrane potential at that particular point. So we're only talking about that small area of membrane where that channel is. So that area of membrane has now depolarized. And here our membrane potential will be around plus 40 millivolts. We've gone right the way through 0 up to plus 40. So we're in a different situation where we are now more positive inside than outside because of that flood of sodium. The inside of the membrane at this particular point is more positive than outside. Let's look at that in terms of our specific tiny area, and imagine we have our specific tiny area with one voltage-gated sodium channel, a voltage-gated potassium channel, and a sodium-potassium ATPase pump. So right at the bottom, we're at resting membrane potential. You can see the dotted line there at minus 50 as threshold, and the top dotted line is depolarization at plus 40. Somewhere in the middle there'll be 0, we don't care about 0. So resting membrane potential in neurons, as we said, is approximately minus 70 millivolts. So there we are, we're sitting in this tiny, tiny section of membrane quite happily at resting membrane potential. We get this adjacent diffusion of sodium. So there is an adjacent rise in sodium that causes the membrane around our little channel to rise to minus 50. As you can see on that red line, it's now reached threshold. So because it's reached threshold, the channel opens. When the channel opens, there is an influx of sodium following its electrochemical gradient. That influx in that tiny area of sodium causes depolarization up to plus 40. So in our tiny area of membrane, we have now depolarized due to sodium influx. Going back to our diagram, this is where we left off. We had our influx of sodium. This is us depolarized. That sodium that has influxed now diffuses out. It diffuses both directions, but we'll explain later how we cope with that. The key thing that we're looking at is to the right there, because that's the direction of our wave of depolarization, our action potential. So our sodium diffuses out, and you can see what's going to happen here. This is taking us back in terms of that sodium channel, the red one on the right. In terms of that sodium channel, we're at the state we were at the sodium channel on the left. We've got an adjacent rise in sodium. That adjacent rise in sodium in terms of that channel further along the membrane has now caused it to reach threshold. Its voltage sensor also has a threshold of minus 50. It's reached minus 50 and it's opened that channel. And the same thing happens here. Then we get an influx of sodium further along the membrane, following its electrochemical gradient, which causes depolarization further along the membrane. This is propagating the action potential along the neuron in the direction that we want it to. At depolarization, the original voltage-gated sodium channel closes. They basically are have a set time that they're open for. And generally that time is fairly standard, and at a point they will close. At depolarization, at plus 40 in this case, they close. At the same time that this happens, the voltage-gated potassium channel opens. Now it's voltage-gated, and it actually got its stimulus from that initial adjacent rise that opened the sodium channel. But potassium channels are a bit slow to open, so they don't open immediately. They delay their opening, and they only open at the same time that the sodium channel is closing, at depolarization, at plus 40. So at plus 40, our voltage-gated sodium channel closes and our voltage-gated potassium channel opens. This time, this is a potassium channel. Potassium effluxes, causing repolarization in that small section of membrane. Remember, it has this strong concentration inside the cell, so it's still following its gradients, still passive transport, and it's effluxing. Now, of course, sodium is a cation, it's positive, so it coming in made the inside more positive. Potassium is also positive, it's also a cation, but it's effluxing. So it's taking that positivity, if you like, out from the cell. So hence it's repolarizing the membrane. Actually, they're slow to open, the potassium channels, but they're also a bit slow to close. So they stay open below resting membrane potential. So they keep effluxing potassium until we get to resting membrane potential, but then they don't shut straight away. They stay open. More potassium effluxes, which takes us below resting membrane potential, and we go into a period of hyperpolarization. Remember, resting membrane potential is polarization. We've just had repolarization to polarization, but now we've dropped below into hyperpolarization. And we'll talk about why that is in a little while. So going back to our diagram then. Remember, this is our one specific original section of membrane where we all started. We got up with sodium influx to depolarization at plus 40. We now know what's going to happen at plus 40. The voltage-gated sodium channel closes. It also has an extra lock. We'll talk more about that in a minute. It locks or gates briefly so that it can't reopen whatever the stimulus. The voltage-gated potassium channel opens, potassium effluxes, causing repolarization right back down to minus 70 RMP, and then drops below, so maybe up to minus 80, minus 90. At this point, our voltage-gated potassium channels close. Once all the channels have closed, our sodium-potassium pumps have a chance to restore resting membrane potential. Actually, they will have been working throughout this whole depolarization, repolarization. But while the channels are open, they've got no hope of correcting that. It's not until all the channels close that the pumps can really be effective in moving 3 sodium out, 2 potassium in, and restoring resting membrane potential. And because of our sodium diffusing along the membrane to the next set of voltage-gated channels, this depolarization, and followed by repolarization, is propagated in a wave along the plasma membrane. It only propagates in one direction because of what we said about the sodium gates. The sodium channels, when they close at plus 40 at depolarization, also gate. So there you can see they've got a little cap on. I always used to say it's a bit like those people that have a front door and then they have a gate across the porch. So you can open the front door, you can talk through it to someone visiting, but they can't get in. There's an additional gate. What this does, it prevents the channel from opening, even if the voltage sensor reaches threshold. Because, of course, we've still got all this sodium floating around and it diffuses in both directions. So if it diffuses back to a previous sodium channel, the risk is that it could take that channel behind to threshold, and if it opened, then we would be propagating backwards. We don't want to propagate backwards. So even if this channel does reach threshold and opens, it's got this little gate for a short while. It's only for a short while, just as long as it needs to be for everything to get cleared away. So it cannot reopen. This is called its refractory status. So when the sodium channels are gated so that they cannot reopen, they are refractory. Another mechanism that we had there was this period of hyperpolarization. Remember, we said the potassium channels don't close at RMP. They continue to efflux potassium. So we go into this period of hyperpolarization. We have moved the situation below resting membrane potential. Therefore, any channels in the area, it's going to be much harder to get back up to threshold to stimulate them to open. These two mechanisms give us the absolute refractory period. So the absolute refractory period, which prevents backward propagation, is when the sodium channels are gated and there is hyperpolarization just before those potassium channels close. So the potassium channels are open, the sodium channels are gated. Okay, so we've just had our action potential coming down on neuron. It's come down and down, down on neuron, and then it has to be transferred into a myocyte. And this happens at the neuromuscular junction. So our action potential is transmitted chemically from the neuron to the skeletal myocyte. The chemical, in this case, is the neurotransmitter acetylcholine, ACH. Always motor neurons, it's acetylcholine. ACH, acetylcholine, chemically transmits the action potential across a gap between our neuron and, in this case, a myocyte, known as the synaptic cleft. The wave of depolarization can then continue in exactly the same way as it did down the neuron. So we've got sodium channels opening, sodium channels closing, potassium channels opening, potassium channels closing, all the way along the sarcolemma, the plasma membrane of our myocyte, once it's been transmitted across the neuromuscular junction. So let's have a closer look at that. Here's our motor neuron coming down. In this case, it will be the lower motor neuron because we're outside of the central nervous system. It has a axon terminal at the end of it. Sometimes in some diagrams you may see it called a synaptic knob, which always gets a bit of a laugh. So we'll stick with axon terminal. So after the axon, we have the axon terminal. This is still the neuron. Then there's a gap, the synaptic cleft. This is just full of extracellular fluid. It's not within a cell. It's a gap between two cells. In this case, a neuron and a myocyte. Over the synaptic cleft, we have the sarcolemma. The sarcolemma is the plasma membrane of our myocyte. And sometimes you will see the membranes on either side of the synaptic cleft referred to as the presynaptic membrane. In this case, that would be the membrane of the motor neuron, and the postsynaptic membrane. In this case, it would be the sarcolemma of the myocyte. So our action potential propagates in the way we've just talked about down the axon of the motor neuron and reaches an axon terminal. Within the axon terminal, there are voltage-gated calcium channels, Ca++ channels. These are voltage-gated, so we have the same situation here. They have a voltage sensor that reaches threshold when our action potential gets to them. They reach threshold, and when they reach threshold, they open. Calcium influxes. There's a lot of calcium outside, not much inside. So calcium, through passive transport, follows its gradient and influxes into the axon terminal. When it gets into the axon terminal, it interacts with the cytoskeleton. Remember the cytoskeleton? Cytoskeletons move vesicles around, they change the shape of the cell, they do lots of funky things, but generally that involves calcium binding to them. So calcium influx is often a stimulus for the movement of vesicles by interacting with the cytoskeleton. So calcium influxes interacts with the cytoskeleton. And this calcium is not to be confused with our calcium that is later released within the myocyte. Remember, we're still in a motor neuron here. That calcium is being released into the myocyte to bind to troponin. Totally different calcium. It's all calcium, but this is a different set of calcium. Here we've got an influx into the motor neuron, into the axon terminal, interacting with the cytoskeleton, and that then stimulates the exocytosis of vesicles of acetylcholine. The neurotransmitter acetylcholine is exocytosed into the synaptic cleft. That acetylcholine diffuses across the synaptic cleft, and on the other side, on the postsynaptic sarcolemma, There are ligand-gated sodium channels. Remember, I promised you at some point we'd have some ligand-gated sodium channels. These are them. Remember, everything else is voltage-gated. This is the only place we've got ligand-gated sodium channels in this process. They're ligand-gated because, of course, the ligand is acetylcholine. Acetylcholine binds to receptors on those channels, specific ACH receptors, on the postsynaptic sarcolemma of the myocyte. Those channels then open, and sodium, of course, will influx. Again, strong electrochemical gradient, sodium always influxes. So sodium has influxed through ligand-gated sodium channels because ACH bound to them. This, of course, is going to cause local depolarization of the postsynaptic sarcolemma. So at the neuromuscular junction, we've now got depolarization within the myocyte at that specific point. This, as always, diffuses out, and next to the postsynaptic sarcolemma, in the rest of the sarcolemma, we have voltage-gated channels. So here we go, this is back to the start of our action potential. Here we can generally call it a wave of depolarization because we're not in a neuron. Sodium diffuses across to the first voltage-gated sodium channel. And you know what's going to happen. That voltage sensor, if it reaches threshold, minus 50 millivolts, will open the channel. So the closest one reaches threshold and opens. And we know what's going to happen now. We get an influx of sodium. So that then gives us depolarization of the sarcolemma next to the neuromuscular junction. And you know what's going to happen next. That's going to diffuse out. It's going to open the next sodium channel along, voltage-gated sodium channel along, and we're back to propagation of this wave of depolarization. But in this case, we're propagating along the sarcolemma. So we have a wave of depolarization along the myocyte sarcolemma. Exactly the same mechanism as we had in the membrane of our neuron, our action potential, same sequence of events, but now we're moving along a myocyte's membrane, the sarcolemma. So we're now back to our myocyte. We had our action potential coming down our neuron, over our neuromuscular junction, transmitted chemically, and it's now waltzing along happily along the sarcolemma. So let's have another look at our myocyte. Remember, we have our myocyte full of myofibrils. These are the specialist organelles that are full of the myofilaments divided up into sarcomeres. Those myofibrils fill the skeletal myocyte. Remember I said about the nuclei getting pushed to the outside, and you can see there a nucleus being squished up against the sarcolemma. Another thing that we have are things called transverse or T tubules. They're green in this diagram. These are invaginations, usually gets a giggle as well, of the sarcolemma. So the sarcolemma basically dips in and forms a tunnel into the myocyte and around the myofibrils. On either side of these T tubules, the sarcoplasmic reticulum, like endoplasmic reticulum, but we call it sarco because that's another prefix for muscle, the sarcoplasmic reticulum, it's known as in this case, forms these pockets on either side of our T tubules. These pockets are called terminal cisternae. If you're talking about one, you're talking about terminal cisterna. These terminal cisternae, they're blue in the diagram. You can see them on either side of our green T tubules. The sarcoplasmic reticulum and these pockets of it, called terminal cisternae, hold a store of intracellular calcium, Ca++. Endoplasmic reticulum in other cells does as well. It's always a store of calcium tucked away in there. Of course, even though it's a cation, in this case double cation, doesn't have any influence particularly on the intracellular environment because it's tucked away inside either the endo or the sarcoplasmic reticulum. So, and bear in mind, this is a separate store of calcium to the calcium we talked about before, just entering the axon terminal. That was extracellular calcium. So, in the sarcolemma, another structure we need to think about are these things, these receptors that are voltage sensitive, and they're found down the T tubules, built in to the sarcolemma that dips down into these T tubules. So first of all, we have these dihydropyridine receptors, DHP receptors. The abbreviation is fine. I don't expect you to remember the full word. DHP receptors are fine. These receptors are voltage sensitive. And then below them, they have rhianidines. So a wave of depolarization traveling down the T tubule. Remember, depolarization, this is voltage, activates our voltage-sensitive DHP receptors. The DHP receptors then activate Rhianidine receptors. It's a bit of a misnomer calling them receptors, I always think. These are within the membrane of the terminal cisternae, those pockets of sarcoplasmic reticulum. The Rhianidine receptors release calcium from the terminal cisternae into the sarcoplasm of the myocyte, like cytoplasm, but in this case sarco prefix for muscle, sarcoplasm of the myocyte. So of course calcium is now available to the myofilaments, specifically troponin, so that it can bind to troponin. So let's put that together in terms of where we left off with our neuromuscular junction. We have our wave of depolarization traveling along from the neuromuscular junction along the sarcolemma of a single myocyte. It follows the sarcolemma because, of course, these potentials are always across the membrane. They have to follow the membrane. Follows the sarcolemma down a T tubule. This depolarization activates the voltage-sensitive DHP receptors, which in turn activate the rhianidine receptors. The rhianidine release calcium from the terminal cisternae, these little pockets of the sarcoplasmic reticulum, and they've released it into the sarcoplasm around the myofibrils and within the myofibrils. So calcium is now free to bind to its binding sites, 2 per troponin. Which brings us to contraction. If you remember, we said once calcium is released, we are at the end of excitation, and now contraction can begin. Excitation, contraction, coupling. Now we're over the hyphen and we're into the contraction bit. And this, of course, happens in the sarcomeres of the myofibrils. If you remember, in every single sarcomere of every single myofibril. It involves, if you remember, we said before, the interaction of myosin and actin. But first, the first thing we had to do was move tropomyosin into the grooves of the helix of actin and away from the myosin head binding sites, exposing them to myosin, those binding sites being on the actin molecules. So when calcium is released from the terminal cisternae of the sarcoplasmic reticulum, because we've activated the DHP, which have activated the rhianidine, two calcium bind to each troponin complex. Troponin then moves tropomyosin into the grooves of actin and exposes the myosin head binding sites on actin. And now we can have our interaction. So there you see we have our actin molecules with their myosin head binding sites pointing towards the myosin. At the moment in this diagram, we have tropomyosin in the way. Below we have myosin. I have only drawn one myosin head because that's for simplicity. In reality, of course, we'd have many myosin heads would be reacting with actin or interacting with actin, and they would be interacting with actin all around the myosin. Remember our 3D cross-section showing us the 3D structure of it. So when our myosin head binding sites on actin are covered by tropomyosin, so this is when the muscle is relaxed, the myosin heads are at rest. Remember I said they're floppy. They're in a low energy state. At rest, because they're floppy, they can't reach up to actin. They also have ATP, a molecule of ATP attached, but it's still ATP. So bear in mind, it's not being used. We haven't released the energy from it. It's like holding a battery, but not actually connecting it to anything. There's no energy unless we hydrolyze it. So it's unused and non-hydrolyzed. And it maintains that myosin head in its current state in this low energy state. But when tropomyosin is moved away by the binding of calcium to troponin and those myosin head binding sites are exposed, ATPase on the myosin heads hydrolyzes the ATP. When it hydrolyzes the ATP, of course, the most important thing is that it releases energy. And this gives the myosin head energy. It's now in a high energy state. It can reach myosin. So it goes from floppy, couch potato mode up to fully alert, high energy, often called cocked. In this state, it can reach up to actin, and because the myosin head binding sites are exposed, it binds to them. So it has used energy from ATP. But it keeps the ADP and the phosphate, the PI, left over from the ATP hydrolysis. So it keeps both components attached to it at the moment. This is the cross-bridge stage. So the myosin heads have used ATP to give them energy to reach up to actin, and they formed a cross bridge with actin. They still have ADP and P attached to them. Once our cross bridge is formed, though, and you can imagine that doesn't really do a lot at the moment. We've got a myosin head reaching up to actin. Nothing has moved other than the myosin head. But at this stage, we do the power stroke. The myosin heads release the ADP and P. That detaches, goes off. This causes the myosin heads to flex forward, and they bend forward while they're still bound to actin. So what they're doing is they're pushing actin towards the M line, just a tiny bit as far as they can reach. But that release of ADP and P gives us this power stroke, where actin is pushed towards the M line. Every myosin head in every sarcomere throughout all of the myofibrils within this single myocyte will move actin towards the M line. So every sarcomere shortens and the entire myocyte will also contract. But this is only a tiny bit that it's moving. So let's see what that means in terms of our sarcomeres in their line. If you remember, in the middle of every sarcomere we have our M line. At the ends we have our Z line. They're just the elastic fibers that enable the sarcomeres to contract and relax. There we've got an orange myosin filament and green actin. And remember, actin has a gap. There's a gap between the ends of actin on either side of the M line. That gap is known as the H zone. In a relaxed state, the H zone is quite wide. The sarcomeres are also quite wide. So this is fully relaxed. But when it contracts, the actin is moved in on either side of the M line. This is going to shorten the sarcomere. The sarcomere becomes narrower, shorter, as does the H zone. Look at the difference in the H zones. This is suddenly much smaller. The sarcomeres themselves, whereas above we've got two sarcomeres there, those two sarcomeres are filling less space in the diagram below. So in contracted sarcomeres, every single H zone is narrower because actin has been pushed towards the M line. And this contracts the whole myofibril and all of the myofibrils within the myocyte. So back to our myosin head. We left him in his high energy state. He's done his power stroke, so he formed the cross bridge. Now he's done the power stroke. He's moved actin towards the M line. But as I said, it's only a very tiny amount. This one movement by myosin, this one power stroke, is not going to achieve a whole lot of contraction, even if it is in every single sarcomere. So they need to do repeated power strokes, depending on the amount of contraction needed. Problem is, he's still bound to actin. So at the end of the power stroke, another ATP comes in and it binds to the myosin head. And if you remember, in this state, myosin is in a low energy, floppy state. So this breaks the cross bridge. So ATP comes in and bound. Myosin head hasn't done anything with the ATP yet. So it's in a low energy state. It's floppy. Actin is still being held towards the M line by other myosin heads. It doesn't ping back. So our cross bridge has been broken. However, as long as those myosin head binding sites on actin are still exposed, so in other words, calcium is still bound to troponin, that myosin head will again hydrolyze the ATP that's just bound to it, and it will give it the energy to form a cross bridge again. Still with the ADP and P attached because we're only at the cross bridge stage. But notice that, of course, it's going to form the cross bridge further back because it's pushed actin towards the M line. So we're now binding further back on actin. So when the power stroke occurs this time, actin is going to get even closer to the M line. So ADP and P come off, we have our second power stroke, actin is moved a bit closer to the M line, and we can carry this on. ATP will come in, bind to the myosin head, put it in a low energy state. The myosin head, if the sites are still exposed, will hydrolyze the ATP, go back into a high energy state, form another cross bridge, but further back on actin. Flip off its ADP and P, do another power stroke, and move it even closer to the M line, until ultimately, actually, the actin filaments can overlap at the M line in fully contracted muscle. So, to go back to the beginning, tropomyosin is moved into the groove on actin because of the binding of calcium to troponin, exposing these myosin head binding sites. Then ATPase on the myosin heads, and don't ask me how the myosin heads know that the binding sites are exposed. I'm not entirely sure anyone knows. ATPase on the myosin head hydrolyzes the ATP bound to it, putting it into a high energy or cocked state, which makes it bind to actin. So we form the cross bridge. ADP and P are then released, and the myosin heads bend towards the M line, still attached to actin, so they push actin towards the M line, narrowing the H zone. Another ATP comes in, binds to the myosin heads, putting them back into a low energy state and breaking the cross bridge between myosin and actin. And if our binding sites on actin are still exposed, we will go round again. ATPase on the myosin heads hydrolyzes the ATP bound to it, high energy state, binds to actin, forming a cross bridge. ADP and P are released, myosin heads bend towards the M line, pushing actin and narrowing the H zone, the power stroke. And then more ATP comes in and breaks the cross bridge by binding to myosin heads and putting them back in a low energy state. And we can keep going round and round and round until our actin has got to the M line. And as I say, in some cases in maximal contraction, it can actually overlap at the M line. So going back to our diagram, remember, in a relaxed state, we have a wide H zone. The ends of actin are a long way away, either side of the M line. In a contracted state, they've moved much closer to the M line. The sarcomeres and the H zones have shortened. And, consequently, our whole myofibril and all the myofibrils within that myocyte will shorten. This will shorten that myocyte. In other words, the myocyte is now contracted. That will have an effect on the entire muscle, and the whole muscle will contract, depending how many myocytes have been stimulated. So as long as calcium remains within the sarcoplasm, and as long as there is sufficient ATP available, remember it has to keep coming in and breaking that cross bridge and being hydrolyzed. So as long as we still have calcium within the sarcoplasm, it can bind to troponin. And as long as we have ATP available to break the cross bridges and be hydrolyzed, this cycle of cross bridge, power stroke, cross bridge breaking will continue. Any repeated action potentials will cause the intracellular calcium levels to rise in waves. And this gives us stronger and more sustained muscle contraction. Remember, the whole thing of contraction depends on the availability of calcium. Graded muscle responses we talked about the number of myocytes excited, so not all myocytes within a muscle will be excited and will contract. Also, of course, you can see there the frequency of stimulation. The frequency of action potentials will determine the amount of calcium available to troponin to keep tropomyosin out of the way. Muscle fatigue, as we said, is avoided by stimulating myocytes asynchronously. This is why if you lift something heavy, you can't sustain it for any length of time because you're using all your myocytes. No one is getting a rest. But there is also the effect of muscles being remodeled. And this, of course, is the concept behind bodybuilding, behind exercise. You can add myofilaments. That causes hypertrophy of muscles. The muscles get bigger. You can add sarcomeres if you stretch muscle. So the myocytes become longer because you put more sarcomeres in. Or you can shorten muscles by reducing sarcomeres. And this really is dependent upon the demands put on the muscles. They respond by compensating. So, time for a bit of relaxation, you'll be glad to hear. So, in order to get relaxation within the muscle, the motor cortex needs to stop generating action potentials and sending them down the upper and the lower motor neuron to the neuromuscular junction. So no new action potentials are coming down. Within the neuromuscular junction, within the synaptic cleft, ACH, our neurotransmitter, acetylcholine, is deactivated by an enzyme called acetylcholinesterase. This basically splits it into two components, acetate and choline. Choline is taken back up into the axon terminal to make more ACH. Acetate is excreted, but the ACH is deactivated. Calcium, this is the calcium in the myocyte that's released to bind to troponin, is pumped back into the sarcoplasmic reticulum via calcium pumps known as SERCA. That stands for, you don't really need to know this, but just in case you're interested, Sarco-Endoplasmic Reticulum, Ca++ ATPase pumps. So calcium is pumped back, it's not available to troponin to bind to troponin. Because of this, tropomyosin then reconfigures, so it comes out of the grooves and it goes back over the actin and blocks the myosin head binding sites on actin. The last ATP that bound to the myosin heads is not hydrolyzed. So if you remember breaking the cross bridge, ATP binds. Normally, if the myosin head binding sites were still exposed, it would be hydrolyzed and off we'd go again. In this case, that last ATP isn't hydrolyzed, so it keeps the myosin heads in that floppy, low energy state. They can have a bit of a rest. So because all this has stopped, the sarcomeres relax, the H zones and the sarcomeres widen, lengthening each myofibril within the myocyte and therefore the myocyte itself, putting the muscle back into a relaxed state. And just to finish off, an interesting point. In rigor mortis, there's two things that happen really. One of them is that calcium leaks into the sarcoplasm and binds to troponin. So because of the state of the body, it just leaks back in from the Sarcoplasmic Reticulum. Binds to troponin. When it binds to troponin, of course, it's going to set the whole process off. Tropomyosin moves. Myosin decides, oh, we're off. I'm going to hydrolyze my ATP, and we're going to have a cross bridge and power stroke, etc., and we're going to have contraction because we suddenly have calcium available. But the problem is, eventually, the myocyte will run out of ATP because there's no oxygen coming in because the person is dead, they're not breathing. So we have hypoxia, there's no oxygen, to make adequate ATP. Remember, most of ATP is aerobic. It needs oxygen. If there's not enough ATP, what will happen is that the cross bridge eventually can't be broken. So the final cross bridge won't have any ATP to come in to bind to the myosin head, so the cross bridge won't be broken. So the sarcomeres, having done a bit of contraction because of the availability of calcium, stay contracted. And that gives rigor mortis. Of course, eventually, after a period of time, the proteins that can't be maintained because of the person's dead, then the proteins actually start to break down. When the proteins break down, nothing can be held together, and that's when rigor mortis ends. The rigidity resolves. Just an interesting point. So that's the end of tissues and muscle, our second topic. Our next topic will be endocrine. But before moving on to that, please consolidate this topic. So make sure your terminology is up to date in your glossary. Make sure you have your own notes, your own diagrams, you've accessed the PDF, and you've attended the live sessions. So there will be a final live session for the second part, and then we will move on to endocrine.